Welcome to video 3.9. In this video, I'll introduce quaternary structure as the last kind of structure that involves proteins for now, specifically proteins that have multiple distinct subunits. So, up till now, we've talked about primary and tertiary structure, but it's worth noting that this is only for single polypeptide chains, right? A single piece of, a single molecule, a single chemical with an N and C terminus. But some proteins are not functional until they associate with other proteins. Now, when I say other proteins, be sure not to confuse this with domains. When we talk about domains, remember this is still a single polypeptide, right? This is one chemical that folds in one region like this and one region like this. These are domains, so this is still tertiary structure. It's a single polypeptide chain that is folding in a specific way. When we talk about quaternary structure, we're going to use the term subunit. What happens with subunits is that we actually have two distinct polypeptides. So one that is red, one that is blue. These are encoded for by different pieces of DNA, or maybe they're the same, but either way, it's two distinct proteins interacting together to form the final quaternary protein. So again, tertiary structure only applies to a single chain. Both of these chains have tertiary structure, but then they come together to form quaternary structure. So quaternary structure only applies to proteins that have multiple distinct polypeptides making them up. Question is, well, what would hold these things together? Right? And the answer is everything that also worked for tertiary structure. Right? The interface between these two proteins is where they want to bind. So we need to make sure we match them in terms of their shape, their polarity, their hydrophobicity. Perhaps they can also be disulfide bond connected, right? So cysteines on multiple distinct chemicals. If there was, for example, a cysteine here and a cysteine here, they could form a cysteine linkage. And now they're technically the same chemical, right? Still came from two distinct polypeptides, right? Also, charges like salts, interactions, lots of reasons why these could want to come together. But again, we call it quaternary structure. And along with that new name comes a new naming convention that we need to learn. So, how do I name quaternary structure? Firstly, we talk about this term Xmer. So it really just depends on how many total subunits there are. So for example, we could have a monomer, a dimer, a trimer, and let's just go crazy, an octamer, for example. Again, mer just means molecule. So we're just saying with this part of the name how many subunits there are, right? Then the other term we need to be aware of is homo versus hetero. For a homodimer, for example, we'd say that all of the subunits, right, so all of the individual polypeptide chains were identical. They were the same to start, right? So for example, if I had, well, I'll cover examples below in a moment. We call it a heterodimer, for example, if all subunits are not identical, if there are different subunits that make up the protein. With that in mind, let's do a couple examples. We'll do these together, and then I'll give you a question below to wrap up the topic. Here we have two alpha subunits, so they're both the same, and there's two of them. We call this a homodimer. Again, homo because they're the same, right? All of 100% of the subunits are alpha, and dimer because there are two subunits. Here, for the next one, we see two distinct alpha subunits and two beta subunits, which means they're slightly different. Just because these are the same and these are the same doesn't matter. If all the subunits are not identical, sorry, that's an important word there, they're not identical, this is now a hetero, and not a dimer anymore, but a tetramer. Right? So note, for example, I could have something like alpha, 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 beta. Right? This would also be called a heterotetramer. At least as far as this course goes, there's no naming distinction between these two, right? Even though they are different. Here we have 3 alpha, 1 beta. Here we have 2 alpha, 2 beta. In both cases, not all of the subunits are identical, so it's hetero, and there are 4 total subunits, so it is a heterotetramer. Feel free to pause the video and work through these 2 practice problems. Great. So for the first one, we can see that there are 1, 2, 3, 4, 5, 6 subunits. So this is going to be, right, a hexamer, or hexamer. Maybe I'll call it a hexamer. Right? And we know that they're different because one of them is beta, so it is a heterohexamer. Yeah, heterohexamer makes more sense. And for this one here, we can see there are 3 subunits, so it's a trimer. All of them are the same, they're all alpha, so it's a homotrimer. And so with this naming convention, we can now handle proteins that are bigger, that are functional when there's multiple peptides that come together to make them. All of these have quaternary structure, but keep in mind that each individual protein is folded based on tertiary structure. Quaternary structure describes the whole, organization of all the subunits. With that in mind, that concludes our protein structure section. In the next section, we'll talk about function, specifically of binding proteins like hemoglobin. I look forward to seeing you there.